Understanding the culture test, a deep dive. The culture test, also known as a microbial culture or bacterial culture test, is a fundamental diagnostic procedure used to identify and study microorganisms such as bacteria, fungi, and viruses. This laboratory method plays a crucial role in various fields, including medicine, microbiology, and environmental science. In this article, we'll explore the intricacies of the culture test, its applications, and the process involved. What is a culture test? A culture test involves collecting a sample from a person, animal, food, or environmental surface and cultivating it in a controlled laboratory environment. The objective is to encourage the growth of any microorganisms present in the sample so they can be identified and studied. The process of culture testing. Sample collection. A sample is collected using swabs, aspirates, or other methods, depending on the source of the specimen. Common sources for samples include clinical specimens like blood, urine, sputum, wound swabs, etc., and environmental specimens like soil, water, air, food products, etc. Inoculation. The collected sample is then inoculated onto a special culture medium that provides the necessary nutrients for the microorganisms to grow. The choice of culture medium depends on the type of microorganism being targeted. Incubation. The inoculated culture medium is placed in an incubator set at specific conditions favorable for the growth of the target microorganism. Observation and identification. After a period of incubation, the culture is observed for the presence of microbial growth. The microorganisms are identified based on their appearance, growth characteristics, and biochemical tests. Applications of culture testing. Medical diagnostics. Culture tests are widely used in medicine to identify the causative agent of infections, determine the most effective treatment, and monitor treatment progress. Microbiological research. Culture tests are essential in microbiology for studying microorganisms, researching new antibiotics, and investigating microbial ecology. Food and environmental safety. Culture tests are employed in the food and environmental sectors to detect pathogens, monitor water quality, and assess environmental safety. Advantages and limitations. Advantages: specificity, versatility, quantification. Limitations: time-consuming, viability requirement, skill-dependent. Conclusion. The culture test is an invaluable tool in microbiology and medical diagnostics, offering precise identification and characterization of microorganisms. Whether you're a healthcare professional, researcher, or simply curious about microorganisms, understanding the culture test and its applications is essential. Various types of culture tests used in microbiology and medical diagnostics are tailored to identify specific types of microorganisms. Some common types include bacterial culture, fungal culture, viral culture, mycobacterial culture, anaerobic culture, aerobic culture, urine culture, stool culture, tissue culture, and environmental culture. Conclusion. The choice of culture test depends on the suspected infection type and sample nature. Proper sample collection, transportation, and storage are crucial for accurate results. Understanding different culture tests is essential for effective diagnosis, treatment, research, and environmental monitoring.